Well, my interest changed as the field changed. When I began to do my research, you measured antibody levels, and that was probably the only really quantitative measure that that one had. And things advanced, and I changed along the way. I guess I should, this may be as good a place as any to say my entire scientific career has been at the University of Chicago, save the one-year internship in pathology at the University of Michigan, and 2 sabbatical years. Now, I don't recommend that anybody stay at the same institution. My own feeling is you ought to do something different about every 10 years. If the first 10 years, you can either do something important or you can make some mistakes. You ought to use the next 10 years to correct the mistakes or do something else different. But I've been lucky at the University of Chicago in that I have had different jobs over the 40 years that I had been affiliated with that institution. So I was able to change jobs about every 10 years, doing something different. I was offered positions as chair of pathology, and I looked at several other opportunities as chair of pathology. But I turned them down because I thought the students at the University of Chicago and my relationship with students at the University of Chicago was the greater attraction. What I did as I had an independent lab was to try to stay active enough in the lab to identify a question that deserved study. And I hope to attract a student. And I've often said the secret to success with graduate students is to select students who are smarter than you. And when a student would come along, I would say, well, I've been looking at this. Are you interested in following up on that? And while he or she was doing that, I tried to branch out and look at a different question in a different way. And that's how my scientific career at the University of Chicago went. I think it was 35 students received PhD degrees on the basis of work that was done, if not exclusively, a large part, in my laboratory. And I had only 7, I think, postdoctoral fellows during that time. Now, I suspect this may be as good a point as any as to say why that was done. I had not had much experience with postdocs until I went on the sabbatical. And there it became clear what should be the goal of a postdoctoral student. In my opinion, at least, as a graduate student, you identify a field that you're interested in, you accomplish something, and you establish an area within that field that you're interested in. Now, postdoctoral training is a given now. And what should that postdoctoral training be? Well, it should be to branch out, to get to learn. Hopefully you've sort of reached the end of something with the techniques that you had learned as a graduate student. And you should seek to branch out and learn something new and go to a lab where that expertise his expertise. Now it's become customary for there to be more than one postdoctoral experience. Now, within that lab, you're supposed to now narrow down your area. And the postdoc next to you is another area and another. And there's more of a competition, as I perceive it, between the postdocs. Each is trying to establish true independence and narrow an area. So I don't find that always a healthy environment. On the other hand, if you have a couple of senior graduate students that are working on different areas, and a new student comes in, there are certain unanswered questions that each of those previous graduate students have, or they need to get things finished up, but the new graduate student coming in needs to learn some of those basic techniques and see enough science going on to know where he or she wants to take. But the senior graduate student should be willing to teach the new student, because he's going to have two hands that are going to become increasingly skilled, and if he's as smart as they both win. They both win. And so I would have probably as many as 5 or 6 graduate students working at various levels, but it was a very collegial environment. And also, these graduate students who, in courses or in social interactions, knew the other graduate students in the institution, knew what was going on. And graduate students are more interested, I think, in helping somebody else exploit this field or this technique. And there was more communication among labs at the graduate student level than at the postdoc level. So that was sort of the general philosophy. We first studied just antibody formation, and then we got involved in what controls the immune response. And I mentioned Don Rowley. He was a graduate of Chicago, came from NIH, joined the faculty, and we worked together. And if you give an antigen, that is something that induces an antibody response, you find that antibodies appear in the blood maybe 3 days after the antigen is injected. They rise up over the next few days, and then they fall quite dramatically. They stay higher than undetectable, but there's this. And so Don asked the question, what do you suppose causes this decrease? Now, is it in fact that the antibody that's produced stops the stimulation by antigen? So what if you give antibody before you give the antigen? And what we found was the antibody response was shut off. So that we got very interested in the effect of the so-called passive immunization, the injection of antibodies made in some other some other animal. What's the effect of this passive immunization on the immune response? And he and a graduate student studied this further in antibody formation, and then they began to turn to cellular immunology, which is the antibody is mediated by molecules. Cellular immunity is lymphocytes themselves carry out the immune function. And what they found out was that if you can induce delayed hypersensitivity, that is the old tuberculin reaction, to sheep red blood cells if you immunize in a particular way. But also that cellular immunity can be impaired by passive immunization, that is, giving antibody before you sensitize. Well, about this time, a surgeon, transplant surgeon, joined the faculty at the University of Chicago, and he was interested in studying kidney grafts in rats. And kidney grafts in rats generate an immune response, as they do in people. So you have to modify the immune response so that the graft isn't rejected. So he came to me, and Don was involved in the other student. Stuart came to me, and we, over the next several years, studied the effect of passive immunization on graft survival in the rat. And we ended up with some promising studies. Incidentally, it was about this time, and Don and I like to think that some of the studies that we did with the administration of anti-sheep red blood cell antigen in the rat influenced the now effective treatment or prevention of erythroblastosis, Rh disease in humans.